everyone. Hello, how are you doing? I hope everyone is doing good. So, welcome to another video of molecular basis of inheritance. So, this is our new chapter. We have already covered Griffith experiment. In this video, we are going to cover Avery, McLeod, McCarthy, Hershey, and Chase experiment. These are two different sets of experiment. Griffith left us with the transforming principle. That is, there was some substances which cause the living R strain, that is a rough strain of Staphylococcus pneumoniae, which was not causing pneumonia in the normal case, but when it was mixed with smooth strain, it caused pneumonia. Okay, so he gave this principle known as transforming principle. Some substance from the smooth cause the R to convert into smooth strain, which causes pneumonia. So now Avery, McLeod, and McCarthy confirmed that what was that substance. Whether that substance was DNA or RNA or protein or something else. So these three, they conducted experiments to find it out. Okay, they expanded the findings of the Frederick Griffith, Oswald Avery, and his colleagues expanded the findings of Frederick Griffith to demonstrate that DNA is a genetic material. So what did they do? Okay, they took that same mixture. Yes, heat killed S. They took heat kill S, and what was their hypothesis? They thought, okay, out of these three, one might be the transforming substance. So they selected three, that is protein, either protein or DNA or RNA. Okay, so what were the three candidates from three substances that they were selecting out? Protein, DNA or RNA. Okay, so they took the mixture or culture solution of heat kill S strain. Okay, normal S strain causes pneumonia. When you heat killed it, it does not causes pneumonia. Then this mixture of heat killed with R type. Okay, then this heat killed S strain was mixed with the rough strain. Now they were testing for what? To see whether protein is the uh, transforming substance, to see RNA, whether RNA was a transforming substance or whether DNA was a transforming substance. So individually they portion out the solution, and individually they tested for whether protein is the transforming substance or RNA or DNA. Okay, so three different setup were prepared. Before that, all the lipids and sugars were removed. Okay, lipids, sugars, everything was removed. Only proteins, RNA, and DNA were allowed to remain in the solution. And then now individually they treated these solution to check whether protein transforms the S in heat killed, sorry, living R into S, or RNA transforms or DNA transforms. For that, they used enzymes. They added enzymes. So to check protein, they added proteinase, which breaks the protein. RNA, they added RNase, which breaks the RNA. To check for DNA, they added DNase, which breaks the DNA molecule. So these solutions were treated with different digestive enzymes. Okay. So step one. Step one, we will consider only protein. Just consider this. So he took the solution. They were the group of scientists. Now, addition of the RNase protein. Okay. After adding this enzyme, what should happen? What will happen ideally? This proteinase will break up the protein molecule. Okay. So if protein is the transforming principle, then the RNA should if what they saw is uh, this caused a transformation, that is the R were converted into S. So this means that there is, because we cleave the protein molecule, right? The protein molecule in the step one, it was broken down. So proteins are no longer there to convert S heat killed, sorry, leaving R into heat killed S. Then the rough strains were added as we just discussed. Okay. So if proteinase already cleaved the protein molecule, so there is no protein in this solution. So ideally transformation should not happen if protein was the transforming material, or that is the genetic material. But transformation did took place. It caused a transformation. This confirms that RNA is not the genetic material. Similarly for RNA. Step 2, sorry, that was RNA. Yeah, that was for RNA. Okay, step 1 was for RNA. Okay, and then similarly for protein and RNA both. So here also what it you can look at the picture. Yeah, if RNase is added and RNA gets breakdown. After addition of the rough cells, what was seen, what was observed, that the S cells appear. Okay, so this also cause transformation, means what? RNA is also not the genetic material. So in both the cases, transformation, that is the R getting converted, the rough strain getting converted into smooth, was observed by addition of proteinase and RNase, which we make sure that protein and RNA was broken down. So what was the other substance that was causing the R to convert into S? So in the third step, in the third category, addition of DNase was there. This removed DNA from the extract, and when it was mixed with the R cells, what was the observation? It did not cause transformation, and hence it was confirmed that DNA is the genetic material. Okay. So the transforming substances could be inactivated by DNA digesting enzymes, but not by protein digesting enzymes. So here we saw no transformation. This gave us confirmation that DNA is the transforming material, it is the genetic material, and neither protein nor DNA. So transformation requires DNA. In the similar set of experiment, to get more proof and more clarity and give a solid confirmation that okay, it's DNA only that is the genetic material, Hershey and Chase carried out another type of experiment. In this, they used different samples. They used bacteria and virus as their sample. They took a virus known as T2 bacteriophage. They took this virus and they were grown in two different setups, one with radioactive phosphorus and one with radioactive sulfur. Okay, now we should be knowing, we have studied in lower standard also. This is the picture of a virus, okay, bacteriophage, a virus which infects bacteria. So the outer body of this bacteria, of this virus, is made up of proteins, and the DNA or the RNA is the genetic material. Now mostly in bacteriophage, the genetic material is DNA. That is why they are testing over here the DNA, whether DNA is a genetic material or not. So DNA or protein. So phosphorus is part of the DNA. We just saw the basic structure. I mean, in the first video, we saw the basic structure of the DNA, that a nucleotide is made up of a phosphorus sugar group and a nitrogen base. So phosphorus will be present in the genetic material, whereas sulfur is a part of the protein. Okay, so the body of the virus is made up of protein, and the genetic material is made up of DNA in case of bacteriophage, which is the name of a virus which infects bacteria. So Hershey and Chase took two different setups. In one setup, they grew viruses in radioactive sulfur, and in another setup, they grew radioactive phosphorus. Sulfur is present in proteins but not in DNA, right? As we just discussed, sulfur is present in proteins but it is not present in DNA. Viruses grown in radioactive phosphorus. So phosphorus is present in DNA but not in protein. So this is the distinguishing factor which helped Hershey and Chase to identify whether DNA is the genetic material or for protein is the genetic material to get foolproof confirmation. Okay, so here sulfur is present in proteins but not in DNA, and phosphorus is present in DNA but not in proteins. This is the fact that you have to be clear with this. Now, what did they do? They took this two different setup. So I told you one setup was viruses grown in radioactive sulfur. Another setup was viruses grown in radioactive phosphorus. So viruses were grown. Then after growing with this, okay, growing means what? So the virus will incorporate sulfur into them, into their body, and your viruses will incorporate phosphorus into the body. Okay, now these viruses were then allowed to infect the bacteria. This is the bacterial cell and this is the virus. So infect means they will inject their DNA material into the bacterial cell here. Okay, similarly here the viruses were allowed to infect the bacteria. So the bacteria will infect means it will inject its DNA into the bacteria. What was the conclusion and observation? So this was the step. Viruses were allowed to infect the bacteria. Here the bacteria is E. coli, and then the virus or bacteria were separated with centrifugation to check which part shows radioactivity. Okay, so the conclusion was that DNA showed radioactivity, whereas protein did not show radioactivity. Have a look at this representation. So in the first batch, wherein sulfur was used. So sulfur became viruses which were growing in sulfur, radioactive sulfur. Sulfur became part of their body. Radioactive sulfur became part of their body. So after infection, that is when the bacteria were allowed to infect the with the virus, and they injected their material inside the cell. They injected the material inside the cell. That is a whatever the genetic material was there. Okay, and the radioactivity remained in their body only. It did not enter the bacterial cell. Okay, it did not enter the bacterial cell. Okay, how? When this solution was collected and it was centrifuged. Okay, when the solution was collected and it was centrifuged, the radioactivity was seen in the upper solution, because the when centrifuge happens, viruses are low molecular weight. The body of the virus is low molecular weight, and bacteria are high molecular weight substances. So the bacteria settles down, the DNA settles down, the genetic material settles down, whatever it is, and the lightweight protein components is floating in the upper part of the solution. So the radioactivity was seen in the supernatant. We call it as the supernatant. So this gave us the conclusion that proteins are not genetic material. Proteins are not the genetic material, right? And then in setup 2, this setup 2, wherein the viruses were allowed to grow with the radioactive labeled phosphorus. This was for sulfur. So sulfur becomes part of the protein, that is the body of the virus. Phosphorus become parts of the genetic material which is inside the head. And then after infection, the genetic material is inside the cell. Here the radioactivity enters the cell. How? This was seen by after centrifugation, radioactivity was seen in the lower part of the solution, that is the pellet, which settles down in the test tube. Okay, so this was the conclusion of the Hershey and Chase experiment, that DNA is a genetic material because phosphorus enters the DNA, which is inside the nucleus of the virus, and the virus infects the genetic material, not the whole body, inside the bacteria. So virus leaves the genetic material inside the bacterial cell. That is how it infects. And after centrifugation, the radioactivity was seen in the pellet. So DNA, not protein, was the genetic material because DNA was transferred to the bacteria. Your sulfur was not transferred. The body was not, body of the virus containing radioactive label was not transferred, and hence proteins are not the genetic material, but DNA is. So that's all for this video. In the next video, we are going to do the packaging in prokaryotic cell, and how this DNA is packed, such a long DNA is packed in eukaryotic cell. Thank you. Take care. Bye bye.